Hello, this is an OCR A-level biology video on Unit 2.5 plasma membranes, looking at the phospholipid bilayer. This is leading on from my previous video on lipids, and before that water and different types of reaction, molecular bonding. Okay, so if you want to watch those videos, you are more than welcome to. After this, I'm going to be making videos on carbohydrates and proteins, but let's just get straight into phospholipid bilayer. So we're looking at plasma membranes or cell surface membrane. Don't just write membrane in the exam, you will not get the mark. You have to either put plasma membrane or cell surface membrane. I know it's tedious, but just do it. It's partially permeable, it separates cell contents from its external environment, regulates the transport of materials in and out of the cell, contains enzymes for metabolic pathways, has antigens to help recognize the cell and itself, and can release chemicals for cell signaling. And you're going to see all those properties and other topics in much more detail and examples and when that's used. But that's just a complete overview of the role of cell membranes. So if I speed it up now, what I'm drawing here is a phospholipid bilayer. As you can see, these black molecules are your phospholipids. You should recognise them from the previous video or previous knowledge. If you don't, you need to read up on lipids before you can really understand this topic. So at the bottom I've drawn the cytoskeleton, and I'm going to label these things for you now. So that's the cytoskeleton at the bottom. You need to understand the structure of eukaryotic cells to understand the cytoskeleton function in moving substances around the cell. Okay. Also, we've got our phospholipids. So in black, we have got our carrier proteins, protein channels, and just peripheral proteins. As well as this, we've got carbohydrate molecules, glycoproteins and glycolipids in between our phospholipids, and they can just be snipped off, used for respiration and other processes. They have all sorts of roles within the cell. Okay. There's one thing I've not drawn on the diagram yet, and that is cholesterol. We'll go into the diagram later, but first of all, let's just look at the factors affecting cell membrane structure, which will include cholesterol. But first of all, look at temperature. So obviously you can have a high temperature or a low temperature. So a high temperature creates high kinetic energy. High kinetic energy causes our membranes to become more fluid. So our permeability increases. A low temperature, however, affects the membrane structure slightly differently. So remember our saturated fatty acids and our unsaturated fatty acids. So saturated fatty acids become more compressed. So when it's cold, they increase the fluidity. Unsaturated fatty acids have that carbon-carbon double bond, and which causes kinks in the tails, which push adjacent molecules away, and they help maintain fluidity. So the proportion of saturated to unsaturated fatty acids determine the fluidity of the cell. So the more unsaturated fatty acids you've got, the less fluid the membrane. Okay. So now let's look at cholesterol. So I didn't quite understand cholesterol before making this video, but I watched this other 1 minute video, and in a second I put in a screenshot from that video showing you the structure of cholesterol, because it is quite hard. Well, I find it quite hard to understand, but I don't anymore. And that's just a point I'm going to make. Don't just watch one set of videos. Don't just watch my videos. If you do, watch loads of other people's, read different sources, because ultimately people make mistakes. I'm going to make mistakes, and people explain it better than I do, and I might explain it better than other people do, but you've got to look at other sources and it really helps you understand and learn and visualize it in the exam. So after my little teacherous rant there, teacherous is not a word, let's get back to the point of the video. So cholesterol is a steroid alcohol which we call a sterol. It has a hydrocarbon ring structure with it attached. It has a polar hydroxide group attached to the end. And if these words seem complicated, the diagram that I'm about to insert makes it so much more easier. They regulate membrane fluidity. Okay, so I think the diagram comes in about now. Right, let's pause it there. So at the end of this molecule, I know it's small, we've got a hydroxyl group, okay, and that is the polar end of the cholesterol molecule. Attached to our hydroxyl group, we've got our sterol, so our steroid section of the cholesterol molecule, and then we've got the fatty acid hydrocarbon tail as such, okay. So it's a hydrocarbon tail, sterol group, and then your hydroxyl group in the end, which is polar. Okay, so cholesterol has a small size, flattened shape, so it can fit within our phospholipid, and I'm going to draw that on after I completely explain it all. It interacts with the phospholipid bilayer, so it reduces the movement of the hydrophobic tails, basically buffers the effect of temperature. So let's see them on this diagram. If you look at the top now, I've drawn bits of green in between all the phospholipids, and that is your cholesterol. So they sit in between phospholipids, prevent these tails from moving, and therefore decrease the fluidity of the membrane. So the more cholesterol you've got, the more resistance to high temperatures and low temperatures of your membrane. So cholesterol buffers the effect of temperature. One thing we need to look at with temperature is proteins, because we know we've got these channel proteins within it, and so far we've really just looked at the phospholipid bit. So I've not made the video on proteins, but most of you already know. High temperature will cause proteins to vibrate, breaking hydrogen and ionic bonds, altering its 3D tertiary structure, and therefore the plasma membrane will fall apart. So if the temperature gets high enough, these proteins denature, the tertiary structure is altered, phospholipid bilayer just falls apart. Okay, and if something falls apart, if it's no longer a plasma membrane, it's no longer a barrier, it's no longer partially permeable, it's completely permeable. It can get completely damaged by ridiculously high temperatures. Okay. Finally, we've finished looking at the plasma membrane. Let's look at some transport methods which you probably will know from GCSE, so I really am going to whiz through it quickly. So first of all, we've got diffusion. So as everyone should know, if you're taking A-level biology by now, diffusion is the movement from a high concentration to a low concentration. And an example of this is aquaporins, and some people might think that is facilitated diffusion. It is not. It doesn't involve conformational change of a protein. Water just flows through these aquaporins. Okay, small non-polar molecules diffuse through phospholipids. So through our phospholipids, small non-polar molecules can just diffuse straight through. That is diffusion. Usually when we're looking at things going through channels, we're seeing conformational changes, and then we're dealing with facilitated diffusion. And you have to really understand the difference. But there's 5 factors you must know affect simple diffusion, and they are temperature. So the higher the temperature, the higher kinetic energy, so that you're going to have higher diffusion. Diffusion distance, the shorter the diffusion distance, the faster the rate of diffusion. So the smaller, the narrower our membranes, the quicker we diffuse. Surface area, the higher the surface area, the higher the diffusion. Size of molecules, smaller molecules move faster and diffuse faster. And concentration gradient, the steeper the concentration gradient, the faster the rate of reaction. The faster the rate of diffusion. So now facilitated diffusion. So polar molecules are insoluble in the lipids, so they diffuse through 0.8 nanometer protein channels. Not all of them 0.8 nanometers, that's just that example the textbook gives me. Large molecules bind to transmembrane carrier proteins and they cause a conformational change in the shape of this protein. It does not involve energy. We're still going from the high concentration to a low concentration, but we're just having proteins facilitate that, so they're helping the movement. Then we've got osmosis, which is pretty much diffusion but for water. It's from a high water potential to a low water potential, and the highest water potential is 0, so all low water potentials are negative. Then we have active transport, which is against the concentration gradients from a low concentration to a high concentration, and we're looking at pumps, so protein pumps, and an example of that could be in companion cells when we're pumping hydrogen ions out of the companion cells. Okay, and that is in unit 3.3. You can watch, I think it's 3.3 or 3.1. You can watch that video if you want. Active transport is the only one that goes from a low concentration to the high concentration, and it involves ATP, which is hydrolyzed to create that energy for active transport. Another example is root hair cells use active transport to transport ions. So finally we've got endocytosis, and that is like phagocytes, they engulf things. Endocytosis is when a segment of the plasma membrane surrounds and encloses the particle into a vesicle. Like phagocytosis, ATP is used in this to form the vesicle. So endocytosis is ultimately vesicle formation, whereas exocytosis is the vesicle fusing with the plasma membrane and releasing the contents out. Okay, so ATP is required for both endocytosis, exocytosis. I'll put that on here if you can even read any of this. I appreciate that my handwriting is poor, but let's just put ATP, and it's used active transport, endocytosis and exocytosis. Okay, so that is the whole of unit 2.5. It's a really short unit, and I've probably extended it a bit too long and made it a bit too hard, but next video we're going to be looking at carbohydrates and proteins, but that will not be out tonight if you're watching tonight, my loyal 9 subscribers. Thank you very much.